Click for IB Biology. Here we're looking at the control of enzyme pathways through end product inhibition of two forms, allosteric and competitive inhibition. Students should be aware that there are other ways to control metabolic pathways through the control of gene expression. This is dealt with in the nucleic acids parts of the course. Here we have a PDF in the description of this presentation, which is down below, and you can use that to make your own notes, and there's a quiz for you to check your understanding. To control the pathway, which is A being converted to B, B to C, C to D, and D to E, with E being what we would call the end product. Each stage has its own enzyme, and of course illustrated here I've put enzyme E1, which converts A into B. So to gain control over this pathway, we've got two methods. One's called allosteric inhibition, and the other is called competitive inhibition, and they both involve the end product E. So as the pathway switches on, the concentration of, so this would be time, the concentration of E increases. At a given level, the concentration of E is able to interact with enzyme E1. If we look at the allosteric interaction, then let's draw the enzyme E1 and give it an active site like so. And E, in this case, will attach to the surface of the protein, but not the active site. This location on which it attaches is actually known as the allosteric site. The attachment of E to the protein structure results in a change in the shape of the protein, which of course is a change in the shape of the enzyme, and in particular in a change in the shape of the active site, and that brings about inhibition. This inhibition is reversible. As this inhibition occurs, the production of E product declines, and also it's being used up for its function. As the levels fall, E is used up for the function, and also this will encourage E to be removed from the surface of the protein, the enzyme, also to support the function. With E removed, the shape is re-established, the enzyme puts back its active site shape, and the inhibition is broken. The pathway switches on again, and this cycle repeats itself. Alternatively, we could have competitive inhibition. Now in this case, the competition is between E the product and A, which is the initial substrate. And of course they're competing once more for the active site. So if we have E and A, both are capable of binding to the active site. In the case of E, however, it blocks the active site, switching off the pathway, and in the same way as previously, the production of E then goes into decline. It's been brought under control. The concentration of E will fall because it's being used up in its function, and so now A has the competitive advantage of being more of them than E, and it outcompetes E for the active site, and the pathway switches back on once more. Both examples are reversible inhibition. Illustrated in graphs, we have increasing substrate concentration here. The normal reaction means that as the substrate concentration is increased, the rate of reaction increases until we reach this plateau point, which indicates that the active sites are saturated with substrate. The rate remains constant at that point, and we have our Vmax. The introduction of a competitive inhibitor, such as what we've just seen, results in the same Vmax, but it's achieved in a more controlled manner. With a non-competitive inhibitor, the presence of increasing substrate has no influence over any Vmax, and this remains significantly lower than it does in the normal reaction or the one with the competitive inhibitor. Below are links to other videos in this sequence on enzyme function, a subscription button, of course, and a link to the playlist so that you can look at the other videos.